Good day to everybody. Today I am going to discuss about bacteriophage. Basically, bacteriophage are virus. So, one type of virus. So, bacteriophage and their lytic cycle. So, today our topic of discussion is bacteriophage or virus. One type of virus is called bacteriophage. Now, when we are going to talk about virus, virus is the smallest and simplest infectious agent. We know that, and it is a microscopic agent. It is smaller than 10 to 100 times smaller than bacteria cell. Bacteria cells are also microscopic, but they are smaller than bacteria cells. So most probably these viruses are the smallest agent present in the earth, and you cannot see the virus with naked eye. Even you cannot see with some instrument like microscope, compound microscope, light microscope, which in the case of bacteria can be seen by this compound microscope, light microscope, but virus cannot be seen. Virus only virus can also only be seen by electron high resolution power and high magnification power electron microscope. So it is very to see the virus, it is very difficult job and delicate job. So common people cannot see it properly, but it's present. We can feel its presence at every time, in every time. Like any disease, so many diseases are caused by virus in case of human beings and the animals and the plants, and also the in the bacteria, case of bacteria. So many diseases are caused by virus, and we have recently experienced the what a smallest agent virus can do, like coronavirus. 2 to 3 years back, whole world was stopped due to this virus, and scared due to this virus. So we although it is very microscopic agent, we cannot see it, but it importance is very it is very important for our life, and we should know about the virus. I have told that virus is a very microscopic agent, and in case of bacteria, the bacteria size is generally 1 to 5 micron only in case of bacteria, but in case of virus, the size will be only 20 to 300 nanometer in case of virus particle. And you can imagine the 1 micron is equal to 10 to the power minus 6 meter, and in case of 1 nanometer it is 10 to the power minus 9 meter. So you can imagine that how small this virus particle is. You can imagine it. So this virus particle is also not considered in a living organism group. So there are so many kingdom is present. We know that Whitaker 5 kingdoms, animal kingdoms, plant kingdoms, Monera, Protista, and fungi. Bacteria all are included in these 5 kingdoms, but virus are excluded, generally excluded from this kingdom, because they are not considered as a typical living organism. They are in between living organism and non-living agents, because when they enters the host cell, attacks the host cell or enters the host cell, they will behave like living organism. They can replicate then, and they have no, their virus particle does not contain any cell organelles like Golgi bodies, ribosomes, mitochondria, nucleus, no thing. Only they contain either DNA deoxyribonucleic acid or RNA ribonucleic acid. Only one can they can contain only one type of ribonucleic acid. So they are generally excluded from the living organism category, but when they enters the host cell, any host cell, they will behave like living organism, but outside the host cell they are actually totally inert. There is no presence of their life. They cannot replicate because they don't have any organelles, and they cannot produce protein, and they don't have the metabolic activities also. So this is the thing. Virus are considered as a in between living organism and non-living agents. So this virus particle actually in this the history of, if we see the history of virus, the the scientist Dmitry Ivanovsky in 1892 first discovered the tobacco mosaic virus, first discovered, and Martinus William Beijerinic, the Dutch biologist, in 1898 first coined the name virus. Actually, this virus is different from the computer virus. Computer virus is the full form of computer virus is the vital information resources under siege. This is a malicious software or malware which will attack the data and files and the software of the computer by replication. So this is a different from this computer virus. It is the biological term virus, and it is discovered by this Dmitry Ivanovsky in 1892, and Martinus William Benjeric coined the name virus. Now we are talking about the bacteriophage. Bacteriophage is we are discussing about mainly bacteriophage. Bacteriophage is a bacterial virus, means those virus are eating bacteria or killing bacteria. They are called bacteriophage. Phage means eater, bacteria eater. Those virus are eating bacteria or killing bacteria. They are called the bacteriophage. And this bacteriophage, Frederick W. Todd, the scientist, in 1915 in England, and sorry, Felix D. Herel in 1917 independently in Frederick W. Todd in 1915 England, and Felix de Horel 1917 in the Paris Institute of France, Pasteur Institute of France, Paris. They independently discovered the bacteriophage, means bacterial virus. Now these virus are very small in size, we know that. So outside the host cell, it is very difficult to control this virus. We know that during the pandemic by the coronavirus, we have seen that it is very difficult to control the coronavirus outside the host cell also. So because they are very small in size, and their structure is so rigid, so stubborn, so it is very difficult to destroy the virus particle. in environment. It is so, and it will be spread through all the environment. So it is very difficult to control this type of virus. And we have seen that by irradiation treatment, ionizing irradiation, most resistance particle organism is the virus. And, because this virus particle needs more than 30 kilo gray ionizing radiation dose to kill the virus particle, whereas the bacteria will be killed, bacterial spores will be killed only 10 to 20 kilo gray. Gray means 1 gray means 1 joule per kg, the absorption of ionizing radiation. This is called 1 gray, and 20, whereas bacteria will be killed, bacterial spores will be killed by 10 to 20 kilo gray. Then whereas the virus particle need more than 30 kilo gray dose of ionizing radiation to destroy them or kill them. So you can imagine the how difficult to kill or destroy the virus particle. Now actually, the International Atomic Society Authority, Atomic Energy Authority, International Atomic Energy Authority has recommended the food irradiation up to 10 kilogray only, not more than that. So you have to cross the limit of the recommendation to destroy the virus present in the any food and anything environment. You have to apply this type of radiation. So this is the fact. Virus are very difficult to. Now come to the classification of virus. International Committee on Virus Taxonomy, IC VT, International Committee on Virus Taxonomy, although divided this virus in some, classified this virus in some groups, but it is not done, it has not been done very systematically like other organisms like bacteria, yeast and mold, fungi, any protozoa, any living organism, animal and plants. They cannot be done like that, because these are actually, they are not at all considered as a living organism. Only they are considered when they are present in the host cell, they are considered as a living organism. They can replicate there. So that's why they are called obligate obligate parasite, intracellular parasite. Virus or particles are called. So the classification and taxonomy system is not so systematic like other living organisms. But although some classification has been done to for bacteriophage also, and another reason is the this bacteriophage, the bacterial virus, the generally molecular biologists are working more working with the bacteriophage rather than virologists. So there is a very little scope to classify and classify the virus particle or bacteriophage particle. Like, although the bacteriophage particle has been classified under the family Viridae. Viridae. So now some group of classification of bacteriophage, I am going to discuss about that. First group is the non-enveloped double-stranded DNA. Those virus particle will call double-stranded DNA non-enveloped. Enveloped double-stranded DNA. Non-enveloped single-stranded DNA. Non-enveloped single-stranded RNA. And fifth one is the enveloped double-stranded DNA. RNA. So these are the main 5 groups of bacteriophage generally present. So you can see this virus will either contain DNA or RNA, deoxyribonucleic acid or ribonucleic acid in their particle. Now I am going to explain this. Before that, I will draw a typical bacteriophage structure, so it will be very easy to understand how this particle will be uh, looking like. So I try to draw a diagram for bacteriophage particle. It is a common type of bacteriophage particle like that. Particular bacteriophage like that, and this is the contractile sheet, and this is the base plate of the, and this is the general the you can tell that this is the actually this is a hexagonal head of virus particle, virus particle. This is the tail of virus. Totally this is the tail of the virus, and it is the tail fiber, and this is the whole thing is the called contractile sheath, and this is the base plate. It is the tail fiber, and this in this hexagonal head there will be some DNA, maybe DNA and RNA are present there, and outside this layer there is a rigid structure, proteinous structure, which is called capsid, and these capsids are made of capsomer and proteomer protein unit, and this is the protein unit outside the hexagonal head, and this is all together called uh, tail of virus. So this is the tail fiber and tail pin. So this is the common structure of the virus bacteriophage particle, and now we are going to discuss about the 2 cycles of bacteriophage. 2 cycles of bacteriophage. One is the most common Lytic cycle. Lytic cycle, and another is the lysogenic cycle. Now I am discussing about Lytic cycle, first cycle, and then after I will discuss about the lysogenic cycle. So what is Lytic cycle? When generally bacteriophage are attacking the bacterial cell, and there are some bacteria cell, and they penetrate their RNA or DNA inside the bacterial cell. Then inside the bacterial or host cell, the DNA or RNA particle will be replicated, and they will form some particle, mature particle, which is called virion, and they will be ultimately lyse the bacterial cell and release from the bacterial cell, and then again they infect the another bacterial cell, like that. So I will, by diagrammatic view, I will trying to explain, I am trying to explain the whole process of lytic cycle. Lytic cycle means the lysis of bacterial cell, which is called lytic cycle, and lysogenic cycle is the another thing. I will discuss about it later. So now, the what is lytic cycle? When we are talking about the lytic cycle are generally divided in some steps. Like first step is the adsorption. Adsorption. Second step is the penetration. Third stage is the transcription. Transcription or assembly. And fourth stage is the lysis and release. So one by one I will discuss about the each and every step. The first step is the in the lytic cycle of bacteriophage is the adsorption. In adsorption, what will be happen? This is the one rod-shaped bacterial cell, and virus particle will attach to this bacterial cell from environment. First step. So there will be DNA or RNA here, and this absorption is very specific. In case of bacteriophage, they will attack, they will absorb, they will attach with the specific type of bacterial strain, because this bacterial cell surface, some lipopolysaccharide in case of gram negative bacteria, and some flagella, pilus, or some protein or carbohydrate are present there, and they are specific to attach with this particles. When means lipopolysaccharide in case of gram negative bacteria, in outer membrane of gram negative bacteria, lipopolysaccharide will be present. So they will be attached with, that's why they are very specific. The specific type of bacteriophage will attach with the bacterial cell, specific strain of bacterial cell. They will not attach with another strain. They will only attach with the particular specific strain. So that's why the virus particle are very strain specific, and it is very important point. So you have to remember this, and then what they will do after that, they attaching with the tail fiber pin, this DNA particle will entered in the DNA particle will be entered in the bacterial cell. When this is called penetration, through the contractile sheath, they will penetrate the DNA, their DNA, phage DNA. Now, when they this penetration of the phage DNA will be happened in the bacterial cell, the bacterial metabolic activity will be ceased by this bacteriophage. And the bacteriophage will take the control over the bacterial cell. All the metabolic activity now will be guided by the bacteriophage genome DNA. So they will seize the bacterial DNA metabolic activity and they will start their activity. So in this case the transcription will be happened after DNA injection penetration. Transcription will be happened. Transcription means you know that production of synthesis of mRNA messenger RNA from template DNA. So the virus template DNA, from virus template DNA mRNA will be produced for replication of bacteriophage. Now is this transcription also divided into three main parts: immediately early part, another is the A1 AB delayed early transcription, and C is the late transcription. In when the DNA particle will be entered in the bacterial cell, or DNA particle of the phage will be entered in the bacterial cell, the bacterial metabolic activity will be ceased, and now they take the control over the metabolic activity of bacteria by the bacteriophage DNA. First, what they are doing, they are producing, they are transcribe some mRNA for their nuclease enzyme. Nuclease enzyme, this nuclease enzyme will degrade the bacterial genome DNA and degrade it, and they will supply by this immediately early transcription, immediate early transcription. They will transcribe the nuclease enzyme which will degrade the bacterial DNA and supply the nucleotide for the production of phage DNA. Now start the phage DNA production will be start, bacterial DNA transcription will be stopped. And then they will also modify the RNA polymerase, to produce the delayed early gene transcription. Delayed early gene transcription, what will we do in this case? Delayed early gene transcription, the cytosine, we know that adenine, guanine, cytosine and thymine, these are the four nucleotide present in the DNA molecule. Now the cytosine molecule will be modified by this RNA polymerase enzyme to produce the 5-hydroxy methyl cytosine in place of cytosine only, and this will be after this will be glucosylated by modified by the glucosylation, then they will be different from the bacterial DNA, because in bacterial DNA will contain only cytosine, but the phage DNA will be produced by the contain 5-hydroxy methyl cytosine, also they are glucosylated, so they will be different from the bacterial DNA. So bacterial DNA, bacterial restriction enzyme will not degrade this phage DNA, and they are modified. So this is called the restriction modification model, and they will not be destroyed by this, and they will produce the phage DNA. Now this one in the also the delayed early transcription, the RNA polymerase will be altered, transcribed to produce the late gene. Late gene means the production of the new phage particle, the head, tail and fiber they will produce, and also the enzyme lysozyme will be produced, endolysin enzyme, which will actually break the cell wall of the bacteria and release the new virion, new mature virus particle, bacteriophage particle, which is also called virion, from this bacterial cell. So many bacteriophage will be released from the bacterial cell, and these bacterial cells are individually then again attack the another other bacterial cells. So this is the process of the Lytic cycle. Actually, by this process they are destroying the bacterial cell, bacterial activity, and they are increasing their number, they are replicating the bacteriophage. But outside the host cell, bacterial cell, they will not do this. So this is the thing happened, and whole process is called the Lytic cycle of bacteriophage. And in this case, when the adsorption is started by the tail fiber here with the bacterial cell, what the time from that to find the time from the release from the bacterial cell, the release from the bacterial cell, the release from the bacterial cell, the bacteriophage particle, mature bacteriophage particle will be released from this there, and this is this time will be actually called, this time will be called latent period. And the how many virus particle, bacteriophage particle will be released from one bacterial cell by lysis, this is called burst size. Burst size, one is the latent period and another is the burst size. So latent period is generally burst size, the number of virus particle released from the bacterial cell. A latent period is the time required to from the adsorption time to the release the bacterial, the virus cell, bacteriophage cell. It is generally 20, the latent period will be generally 25 to 40 minutes time required, and burst size will be generally 29 to 133. So one bacterial cell will release 29 to 133 bacteriophage particles, but one bacterial cell, and this 29 to 130 bacteriophage, new bacteriophage will again attack or the other bacterial cells. So this is today, this is enough for today. Next time I will discuss another topic.